Okay, I'd like to thank everybody for joining us today. This is Jeff Bodycomb with Horiba Scientific, and today I have the pleasure of introducing and hosting Dr. Rob Lee with Pharmaceutical Sciences in Bethlehem, Pennsylvania. Dr. Lee has over 20 years experience in the pharmaceutical industry and is currently now the Vice President of Pharmaceutical Development at Particle Sciences. Today he's going to talk about a pair of Horiba technologies. The first is particle size determination, and the second is Raman spectroscopy. And the title of his talk is Particle Size Determination and Raman Spectroscopic Evaluation of a Semi-Solid Vaginal Dosage Form. With that, I'd like to turn the virtual floor over to Dr. Rob Lee. Thank you.
Okay. Well, I am happy to be here. Jeff, thank you very much for the invitation. What this presentation is a follow-up of the same presentation I gave during Horiba's open house a bit earlier this year. And Jeff and a couple of the other my Horiba colleagues asked me to repeat that, so I was delighted to do that. So what I'm going to do is spend a few minutes just to give you some background on myself and particle sciences, so that'll give you a bit of a basis on how we generated this data, how we got into this area, and then give you some of our results on particle size analysis using both the Horiba LA-950 and image analysis using the Climic software for sizing a particulate system in a gel, and then talk a bit about some Raman spectroscopic data that we're generating on our drug-eluting devices. So first off, my name is Rob Lee. I'm VP of Pharmaceutical Sciences at Particle Sciences, and I've been in the industry about 20 some odd years. Got my PhD in Santa Barbara and went through a series of companies including Sterling Winthrop, Nanosystems, Elan, some small drug delivery companies. I was previously at Novavax prior to joining Particle Sciences about 3.5 years ago, and I've been in several areas of product development, so I am also intimately familiar with particle size analysis, having started Nanosystems Analytical Sciences group. So a bit about particle sciences. We don't do any discovery chemistry. We are a CRO providing CMC support. Our sweet spot, as you might guess, is BCS Class 2 molecules. By far the majority of APIs that we work on are BCS Class 2s. We have a lot of characterization, physical characterization methodologies and techniques, a lot of instrumentation. We are very good at formulation development and drug delivery, and have full analytical and clinical trial manufacturing of both sterile and non-sterile products that would help our clients help support our clients' development programs. And some areas that we are continuing to grow is we have a lot of interest in our capability of handling highly potent compounds. So we could handle SafeBridge, up to SafeBridge Class 4 molecules, and we have withstood scrutiny by both large and small pharma. And so we're continuing to grow in that area. And we also really focus on niche drug delivery technologies. So with respect to the company, we've been around for 30 years. We're about an hour from both Newark Airport and Philadelphia Airport. We really do have a well-equipped physical-chemical characterization lab and also analytical labs. And we are DEA licensed, we are registered with the FDA, and we have all the other support for our clients' development programs. As far as our staff, we have a fairly experienced staff. We have a lot of creative people that like to invent things. We have well over 100 patents, not all at Particle Sciences, but a lot of that IP has gone on to be used in commercial products, so it's not really just esoteric things that our folks are interested in developing, but actually things that can be quite useful and commercially viable. So what we do is, as far as drug delivery technologies and approaches, one size does not fit all. We spend a lot of time with our clients to try and understand their issues, their concerns, and exactly what they're trying to get out of the development programs, and we are able to provide them, hopefully, with the best solutions, and not just one solution, but in some cases 4, 5, 6 different drug delivery approaches. And so how we do this is, we do believe one size does not fit all. We look at in-licensing a lot of drug delivery technologies. We have proprietary technologies that we have invented. We have a lot of non-proprietary technologies, but in any case, we leverage the different methodologies irrespective of the route of administration and apply it to a client-specific project or program. And one of the how we continue to grow is not necessarily by adding headcount, but by forming strategic alliances with companies that are that have the same philosophy and have complementary technologies. And we also like to partner with instrument companies, including Horiba, that we feel have what represents best in class with respect to the equipment that they manufacture and sell, and also API manufacturers, excipient manufacturers that have services or products that could be of value to our clients. So what we do, by far the majority of our projects that we work on are sterile products, mostly small molecules, mostly preclinical, early clinical. There are a few of our clients that are kind of guiding us or are requesting that we be their commercial manufacturing organization. We will consider that on a case-by-case basis. And as I mentioned before, the bulk of our compounds that we work on are VCS class 2, which are the molecules that have poor aqueous solubility but are membrane permeable. So that pie chart down at the bottom gives you an idea of the solubilities of the APIs that we've worked on in the last couple of years. So I mentioned earlier that we do not do any discovery work here. We do have a few good medicinal chemists, but that is not really our strong point. So we get molecules post-discovery, and we could take them through commercialization, and we could do all the intermediate steps with respect to CMC, such as preformulation, excipient compatibility, analytical method development and validation, preparation of GLP and GMP material, and so on. And we could also offer our clients bioanalytical support. So with respect to our clients, we have a fairly balanced portfolio, not-for-profits, government, large and small pharma. And with respect to our expertise, I mentioned that our forte is drug delivery. As our name implies, we do a lot with particulate-based systems, micro and nanoparticles. But since we deal with a lot of BCS class 2s, we do quite a bit with solid solutions. And we have a really growing program in drug-eluting devices such as intravaginal rings and IUDs. So just a bit more. I mentioned these before, so I'll just go through these rather quickly. I just want to give you an idea of the type of equipment we have. We have microfluidics, pure nano systems, a couple microfluidizers, a Lystritz 18mm twin screws. So we are well equipped. And as far as analytical tools, we have several different. These are more typical analytical tools: HPLCs, UPLCs, mass specs, thermal gravimetric analysis, etc. Physical chemical characterization, we're very adept at particle size analysis and zeta potential rheology, etc. So now on to the meat of the presentation. With respect to intravaginal dosage forms, this slide gives you a snapshot of the different dosage forms that are applied vaginally, intravaginal rings. As I mentioned, we have a really robust program in developing and scaling intravaginal rings that elute either hormones for contraceptive or microbicides for HIV prevention, or in a couple cases a combination of both. We have done a lot with gels, and I will be talking specifically about a semi-solid aqueous-based gel that we were interested in, where the API was not soluble, so it was a particulate, and that brings up a lot of different questions, or some questions as far as how do you release these gels. So you would think that you would have to track the particle size at release and also as a function of storage. And so there are different methodologies to determine the particle size of particulates in a semi-solid. It is challenging, and the question always arises, how can you validate those methods? So we have been able to develop two validatable methods, one based on laser diffraction and the other based on optical microscopy. With respect to the gels, they are a viable dosage form. It allows the women in third world countries to protect themselves without the knowledge of their partners, which is really a key. These dosage forms can contain one or more APIs, and as I mentioned, some of these APIs are not water soluble, and so they are particulated, and we have to make sure that the particle size does not grow as a function of storage condition. So we looked at laser diffraction, optical microscopy. The two techniques offer both have pros and cons. With respect to the laser diffraction, it's fast, it's robust, it's an ensemble methodology, so you are able to sample millions of particles. With light microscopy, some folks think that that is a gold standard, gives you data on both the particle size and morphology, and it is a count technique, so it is limited. In our case, we sampled 10,000 particles, but you can automate the system and sample up to 100,000 or more. But it is considered the gold standard, but both methods will give you different data, because in the case of real-life samples, they are not spherical necessarily, so if they have any aspect ratio or any irregularities, you are not going to get the same particle size information from both techniques. Okay, so a bit about the laser diffraction methodology. With respect to how we develop the method, we use a paste cell. We use two polystyrene standards that were spiked into the placebo gel, and we varied the sample thickness between 100 and 250 microns, and we're targeting a 90% transmittance level. What we're looking at with respect to the data that I'll show in the next several slides is the D50 on a volume basis. And one word of caution, with these semi-solid samples in a paste cell, the issue that we always run into is bubbles. A lot of the sample preparation goes, and actually the sample preparation time goes to developing methods on how you could minimize the bubbles. And so what we did was, once we were able to work out the sample preparation issues, then we looked at a couple gel types. One was clear, and the second sample was kind of a translucent, hazy gel. So we wanted to make sure that in both cases we were able to generate meaningful data. So for both gel types, the placebo A, which was the transparent gel, and B, which is the hazy gel, you can see that the the actual versus determined size is quite, the fit is quite good for both gels. With respect to precision and accuracy, you can see that from the RSDs for the precision samples, these are replicas of 3 or 4 for the given particle size. The RSDs are satisfactory. With respect to the accuracy, we looked at a variety of particle sizes, and in the case of both gels, the accuracy was acceptable. So what we were able to do is we could show that the particle size of polystyrene standards can be determined quite accurately and precisely in an aqueous gel with both clear and translucent, hazy gels. As I mentioned, the sample preparation is really critical. The key point is to minimize or eliminate the bubbles. The response for the styrenes was extremely linear with very good RSDs. So moving on to light microscopy. We looked at two APIs and an aqueous gel. The first API was soluble, the second API was insoluble, and this method was developed to monitor the particle size of the insoluble API on stability storage. We used an optical microscope coupled to Climax image analysis software. We were looking at 400-fold magnification, and one thing to note is that for optical microscopy, you're already looking at particles north of a micron. So anything below a micron, you might be able to see it depending on what kind of filters or other equipment you're using. But for purposes of our analysis, you're really limited to particles over a micron. So a bit more about the particulars of this experiment. We looked at 300 fields, and we were looking at counting 10,000 particles. It really is an overkill in the case of a polystyrene standard because of the very low the unimodal distribution of the standards. However, for real-life samples that have a broad distribution and that are not necessarily unimodal, you would have to get to at least 10,000 particles in order to have some statistically meaningful analysis. So with these samples, we held the sample thickness to set 25 microns, and we looked at the D50 both on a number basis and a volume basis. So for linearity, this was the average of 3 experiments, looking at the number mean, the volume mean, and the number median and volume median. You can see that for the most part, the number and the volume values are in agreement. And there was a bit of a, in the case of the 25 micron particle sizes, the number means were a bit on the low side. But in any case, the data looked reasonable. With respect to accuracy, you can see that once again, the median values are a bit better than the mean values for in the case of the larger particles. But in any case, it's not really accurate. But I think for this type of sample, you could it is useful for generating the data. So with respect to the precision, here are the data. The precision value standard deviations are quite reasonable, except in the case of the 25 micron particles again. So in conclusion, the linearity for all these samples are quite acceptable. There's good precision, no RSDs greater than 4%, and reasonable accuracy. So looking at actual samples, we have 4 gels, and they have the following data. Number means ranging from 2.7 to 8 micron, and also from a volume basis from 5 to microns. For gel D, the sample precision for 6 replicates are given below, and in the case of the volume mean, which is the highest RSD, it's still below 4%, so that's reasonable. So comparing light microscopy with laser diffraction, how we did that was we spiked White House glass standards into placebo gel, and we used both the Climax image analysis and the LA-950. So why we use these standards is the White House standards are polydispersed, so they range from approximately 1 to 10 microns. We did the standards at 0.1% on a volume basis with placebo A gel, and then we analyzed the Climax image analysis by exporting into Excel. And we also did the same thing for the Horiba paste cell. So this slide is a bit busy, but looking at the number distribution for the 2 methods, the lines with the triangles, squares, etc., represent data based on the laser diffraction system, and the solid lines represent data for the light microscopy. And for the most part, they overlay for the two methods with respect to a given gel. This is another way to look at the data. So it gives you an idea that the cumulatives and the actual the Gaussian or the distributions are for the two methods are consistent. So if you look at the average data, the cumulative data for the replicates, you can see that they overlay quite nicely. So both methods do give comparable data. So in comparison with the different gels, you see that the for gel A, the light microscopy versus Horiba are in excellent agreement. And in that sample, the aspect ratio was on the order of 1.5 for the API. Gel B, which has the highest discrepancy between the light microscopy and the laser diffraction data, light microscopy being about 20 micron versus 50 micron for the laser diffraction data. In that case, the API had the highest aspect ratio of 2.4. So it seems like the potentially the aspect ratio might have might create a bit of a difference in the or discrepancy in between the two methods. So in conclusion, both the laser diffraction and light microscopy with image analysis are suitable for measuring the particle size of the particulates in the gel. You can use them to assess the stability of the formulations and storage. They give comparable particle size distributions both on a volume and are on a number basis, and both techniques are capable of being validatable or are validatable. So shifting gears a bit, we have been doing quite a bit of work on our microscopic Raman system, which is the Horiba Explorer. And one of the things that we're really interested in working towards, and actually we are collaborating with Horiba on this, is ideally we would like to help them integrate the image analysis capabilities of the Climax software with the Raman capabilities of the Explorer. So in other words, we could use the Climax image analysis to look at a field of particles. We could generate the morphology and the particle size data on a sea of particles, and then go back and look at the individual particles and generate the Raman spectra of the particles, so we could determine which is in a sea of particles, what's API 1, API 2, what are the potential degradants, what are the excipients, and really track how these particles grow as a function of stability storage conditions. So a couple things: we could get a very good idea of content uniformity, and also a very good idea of the physical stability of the different components within a semi-solid containing particulates. So with that, here's a typical field of our gels. These are different particles. You can see some are rather transparent. Some of the particles are quite opaque. So the first step, you record an area for your analysis. You look at the region of interest, and that's blown up on the right-hand side. And as I mentioned, you can see the particles are, in some cases, aggregated. You see some transparent particles. You see some that are very opaque. So it does clearly look like there are at least two different species in this system. The next thing you do is you measure the Raman spectrum in an XY map, and we used a 1 micron step, and then we select a characteristic Raman band and map its intensity. So, for example, if you look at the Raman, the single spectrum, you see this peak that is centered at about 2250 or so inverse centimeters, and so we use that as the selected wavelength, and then we generate this map on the right-hand side. So that's an intensity map. So looking at that peak, the dark areas indicate areas where there's a strong signal due to that single peak in the Raman spectrum. Next, what you do is This is a little bit of a busy slide. The particles on the left-hand side, the particles in blue are selected by image analysis and confirmed by Raman as not being API. Okay, so if you look at the clear particles, we selected the clear particles, and if you look on the right-hand side where you're looking at the Raman intensity, you can clearly see that there is no Raman signal corresponding to those particles. Those particles on the left-hand side that are in the green circles were selected by particle analysis as the API and also confirmed by Raman as being the API. In the third case, the particle in the red was not selected by particle analysis as API, but shown to be API by Raman spectroscopy. So you can see the utility of this. You can look at the different particles. It really does give you complementary information compared to what you could get off straight image analysis. And actually, shifting gears a bit, we do quite a bit with drug-eluting devices. This is a cross-section of an intravaginal ring, and it's approximately 4 millimeters in diameter. One of the questions that we always have is we deal with a lot of different APIs and a lot of different solubilities in the thermoplastics that we're working with. And so typically we work with either ethyl vinyl acetate or polyurethane as a thermoplastic, and we incorporate 1 or 2 APIs that may or may not be soluble within the thermoplastic. And the question always arises, what is the content uniformity of the API ring? Does it change during processing? Does it change during storage condition? Does it change as a function of our elution testing? So in all the cases, in our development cases, how do we look at the elution profile, how the API gets out of the polymer? And this is analogous to dissolution testing for solid dosage form, but in the case of an intravaginal ring, the ring is not erodible. Therefore, we're looking at the elution of the API from the ring. And so that without Raman, we could generate quite a bit of data, but it is really gratifying to look at the rings themselves and confirm some of our hypotheses. And we're doing a lot more work in this area. These slides are several months old. We have a lot of recent data showing some rather interesting data, but I did not incorporate it into this presentation. So with that, this is a cross-section of an ethyl vinyl acetate intravaginal ring. The square in the blue is a region of interest. And if you look at the Raman signal from a peak that's within the ethyl vinyl acetate spectrum, you see this green color that's uniformly present within the ring. And so it should be, the ring consists of mostly ethyl vinyl acetate, so you should have a uniform signal based on the Raman signal for the ethyl vinyl acetate. This is the Raman signal for a peak in the API spectrum. And so once again, you can see that the API is uniformly distributed within the ring. And if you really look at it, it looks like, on the right-hand side, you might think that the intensity is tailing off, but that's not necessarily the case. Just suffice to say that this is really showing that the API is rather uniformly distributed within the ring. So if you then look at the ratio of the EVA Raman signal to the API signal, you'll see that once again it is rather uniformly distributed within the ring, and it looks like it's a little bit less intense in the middle, but that's for our purposes, actually, that it's essentially showing that the EVA API signal is rather uniform within the ring. And so with that, I will conclude, and I'll be happy to answer any questions.
Okay, great. Well, thank you very much, Rob. And it's very interesting. Let's see if, yeah, so this is a good time. If you have questions, you can type them into your question or chat box, much the same way you responded when I asked for the microphone check. And so, okay. And now, yeah. Rob, you mentioned, I have a question. You were comparing light microscopy and laser diffraction for your unknown sample. So you talked about using the polystyrene latex and the White House steroid. And you said some of the difference between the diffraction and the microscopy was due to shape. Is it possible that the width of this unknown sample size distribution also affected matching? That is, if the distribution was particularly broad or narrow, you would get, or I should say particularly broad, you would get a different answer by diffraction compared to microscopy?
Yes, I would think that there's a lot of factors that could play into the discrepancies between the two techniques. As you get really into particle size analysis, you could generate a number, but what does that number really mean? You really have to understand how that number is derived. And so in the cases of some of the data I was showing, it might look, if you're used to seeing chromatography data, you might think that the data are really very, very different. But on the other hand, if you look at the entire data and how it's being used, it is not, the data are pretty consistent.
Oh, okay, great. Well, I hope that clarifies things. And we have another question coming in. Is it possible to reconcile elution data with Raman data for the vaginal rings? So I guess what they're asking is you have the Raman map of the vaginal ring cross-section, if that kind of lines up with what you see in your elution studies.
Good question. There are several different, or not several different, but there are different mechanisms that you could think of with respect to the elution of an API from a ring, such as does API be eluted more quickly from the surface of the ring, and then that creates gradient, and then within the ring you have the API from the center migrating to the outer portion of the ring at a different rate. There's a lot of different mechanisms you could possibly think about based on the diffusion of the API within the ring, and then the subsequent release of the API from the ring itself. And you could do mathematical models of those, and then look at the elution profile. And in some of the cases that we're looking at right now, based on the physical chemical properties of the API, such as solubility within the matrix and the size diffusion coefficients within the matrix, we could actually map how the concentration of the API is changing as a function of storage in the elution media. So you could start seeing in some cases that you are depleting the API from the surface, and then you're creating a gradient within the ring. And whereas in some cases you might have a much more uniform distribution of the ring as a function of storage in the elution media. And so we are starting to get a lot more insight into the mechanism of how the API actually is transported within the ring into the elution media. And then from there, ideally draw some conclusions with respect to how that relates to in vivo activity. But one thing that's very strange about intravaginal rings is that for some of our studies, we have some published data on the PK profiles of the API for certain devices, and so we designed our elution media to try and match the in vivo PK levels. And what we have seen is that there really is not much correlation between the in vitro elution profiles and in vivo performance. By that I mean that the vaginal environment seems to be an extremely good, has a lot of solubilizing power. So a ring that might have a very water-insoluble API that you would not think would be very bioavailable, once you get into the vaginal environment, you can see that, and the API is well dispersed, and the bioavailability is a lot higher than you would predict. And so I would caution folks for trying to draw too much from in vitro into the in vivo performance.
Wow. Okay. Yeah, because it looks like the Raman data could help to inform the mechanism, the models you generate for this elution from the ring. So that
Right.
looked quite interesting. Okay. Well, we haven't had any more questions come in. Okay. Well, I suppose that I'll kind of wrap this up unless someone goes ahead and types in a question. I'm going to send everybody our website, www.horiba.com/us/particle. And I'd like to thank everybody for their attendance. Rob, your website is particlesciences.com. Is that correct?
That is correct.
Okay, let's go ahead and type that into everybody's chat box.
And if anyone has any questions, I would be happy to address them. My email address is rlee@particlesciences.com.
Okay. Okay, so I'll go ahead and type that into the chat box as well. My email address is jeff.bodycomb@horiba.com. And then, as you can probably gather, Rob and I talk a lot, so if you find one of us, you've effectively found both of us. So with that, I'm going to go ahead and end this webinar. If the software works correctly, there will be a few poll questions at the end, which is really a way for you to tell really tell my boss how well we're doing with this effort. Okay, well, thank you all for your time, and have a good afternoon. Bye-bye.